In this video, we're going to discuss nucleic acids in terms of our biochemistry option for chemistry. First of all, we'll explore what a nucleotide is, and that's the basic unit of a nucleic acid, and it's composed of three parts: a phosphate group right here, or a phosphoric acid, a pentose sugar, in this case it's ribose, and then a nitrogenous base, or a nitrogen base based. These molecules are combined through condensation reactions. So you can see that the phosphate joins onto this carbon right here. That's carbon number 5 in the pentose sugar. So 1, 2, 3, 4, and then 5. And then this second condensation reaction happens between carbon 1 and the nitrogenous base. So remember, when you're combining or showing the combination of the components that make up a nucleotide, you will need to show that 2 water molecules are involved. So the phosphate is right here. It comes from phosphoric acid, which is a weak triprotic acid. So there are 3 protons that can be donated, and then a double bonded O here. We represent it when we're drawing simple nucleotides as a circle with a P drawn in the middle. The second component is a pentose sugar, and the difference between the 2 different pentose sugars, ribose has an extra oxygen. So deoxyribose on this second carbon, so this is carbon 1, we always go clockwise around from the oxygen. On carbon number 2, ribose has a OH. On carbon number 2 for deoxyribose, it's just a hydrogen. So that's how they differ. And the final and third component is the nitrogenous base. There are 2 bases that are known as purine bases. Those are adenine and guanine, and they have 2 fused rings, so they're right here. Thymine, uracil, and cytosine are pyrimidine bases. They only have 1 ring. All of these structures are found in section 35 of the IB chemistry data booklet. So you don't need to memorize these structures. So we've discussed how a nucleotide is prepared through condensation, but different nucleotides are also combined through condensation reactions. So what happens is the hydroxyl from carbon number 3 of the first nucleotide interacts with the hydroxyl on the phosphate that's attached to the 5th carbon from the second nucleotide, forming a dinucleotide and 1 water molecule. And these bonds are called phosphodiester bonds, and they're covalent bonds. So our cognitive textbook has a helpful exam tip. So when you're balancing equations for condensation polymers involving nucleotides or carbohydrates or amino acids, etc., when you're forming a polymer, the number of water molecules that are produced or evolved is always 1 less than the number of monomers. So if you join 6 nucleotides together, you would have your polynucleotide plus 5 waters. If you joined 3 monosaccharides, you'd have the trisaccharide plus 2 water molecules. Now we'll explore DNA and RNA. So RNA is a single-stranded molecule, whereas DNA is double-stranded. And with DNA, the strands are said to run anti-parallel to one another. So in one strand, the phosphates are pointing up. So that top, wherever the phosphate is pointing up, is called the 5' end. We'll actually draw that on. So up here is the 5' end. Down here is the 3' end. And then this one would be 3' and 5'. So the phosphate, remember, is added onto the 5th carbon in the ring. And then the second one is joined onto the 3rd carbon of the sugar. And again, you should be very familiar with this already, but adenine pairs with thymine. It actually forms 2 hydrogen bonds, and then cytosine pairs with guanine and forms 3 hydrogen bonds. This is just a more detailed view showing where the hydrogen bonding is occurring. So if you see here, this carbonyl is interacting with the hydrogen on adenine. And then this right here is interacting, so this hydrogen is interacting with that nitrogen in the ring. So there's where our two hydrogen bonds are forming. And then over here, this hydrogen on that group is interacting with the carbonyl. Again, nitrogen, hydrogen, nitrogen. So there's a hydrogen bond there. And then the other carbonyl down here interacts with the hydrogen attached to the nitrogen. So cytosine and guanine have 3 hydrogen bonds, and this one has 2 hydrogen bonds. So again, a very familiar structure for DNA is the double helix. And the reason why it forms a double helix is, first of all, the 2 strands hydrogen bond to one another, but then the sugar phosphate backbone is hydrophilic. So it wants to interact with the aqueous environment, but the nitrogen bases are hydrophobic. So they hide inside the structure and they get attracted to each other, and that makes the structure twist. Ribonucleic acid or RNA doesn't tend to interact for very long, so it does not form the helical structure. It may fold and form into tRNA, and the messenger RNA that's produced during transcription just travels to the cytoplasm and is read and translated very quickly, and then is digested. Again, in our biology class, we learned how DNA is packaged. So to protect the structure, DNA is actually wrapped around histones, which are 8 stabilizing proteins, and they wrap around to form a nucleosome. And then the nucleosomes coil further to form chromatin fibers, and then they coil on each other to form the visible chromosomes that we recognize. And the histone proteins are positively charged, and they get attracted to the negative phosphates in the backbone. So we didn't in biology learn why they're attracted to one another, but it's because of that positive charge on the histones and the negative phosphates in the backbone of DNA. Again, DNA is the genetic material, so every nucleus has the entire genome for the organism, so all the instructions for building a new organism. And before cells divide, that genetic material needs to be carefully duplicated. This allows the genome to be preserved from cell to cell. So the process of making 2 new cells is called mitosis. So it's dividing the parent cell into 2 genetically identical daughter cells. In biology, you learn the details and the steps of that. We won't do that for chemistry. In the process of replication, that's the process of doubling the DNA so that the 2 new cells can have a full complement of DNA. So what happens is DNA will unzip only a little by a little because you don't want it to get tangled or break apart. So we break the hydrogen bonds. We don't need to know the enzymes for chemistry. You just need to know that there are enzymes involved, and they only act on small sections at a time at the replication fork. We also won't learn about the leading and lagging strand, but in biology we learn how the enzymes can only work in one direction, so then there's a leading and lagging strand. And what happens are free nucleotides that are floating in the nucleus will pair up with the complementary bases on the single strand. And you can see that happening here. So this thymine has attached to adenine, guanine to cytosine, and so on. And when the DNA is unzipping, that's, again, the hydrogen bonds are breaking, but the actual covalent bonds in the backbone don't get altered. The model of DNA replication that we support and understand is called the semi-conservative replication model. So what happens is, just as I've described, the DNA helix unzips and then new nucleotides are added to the single strands so that the final product will have 1/2 the original DNA and 1/2 the newly synthesized DNA. In transcription, that's the process of making an RNA copy of a DNA gene. So we're not copying the entire genome, just one part of it. So this again, will need to be unwound in the section that needs to get copied. And then RNA polymerase is the enzyme that catalyzes the production of the new strand. And then when the RNA is made, it travels to a ribosome in the cytoplasm to be translated. In translation, the mRNA that's been transported to the cytoplasm is converted to a protein, and the code is read in triplets, and those triplets are called codons, and there are 64 possibilities for the 20 amino acids because there are 4 different bases and an equal possibility of using any of the 4 bases for the codon. So there'd be a 4 times 4 times 4 possibilities. So 64 possibilities. Some of these possibilities are actually stop codons. So you can see there's one here, UGA. Another one is found here. So UAA and UAG. So those are the 3 stop codons. So AUG is the start codon, which is found here. So AUG is methionine or MET, and that's the first amino acid in every protein. Now, interesting, you don't have to use the genetic code for the IB chemistry. You don't have to be able to read a codon chart, but of course in biology you do. So translation happens with the help of tRNA and the ribosome. So the mRNA is threaded through the 2 ribosome subunits where the code gets read, and tRNA brings the amino acid that matches the codon to the growing polypeptide. And when the ribosome reaches a stop codon, translation will stop, and everything will fall apart and the protein will fold into its structure. So there are mutations sometimes. So sometimes errors are made in the DNA. And if they occur in regions that don't code for proteins, the mutation is harmless. That's called a silent mutation. But even sometimes a single base pair can cause huge effects, such as in sickle cell anemia, which is one of the examples you need to know for your biology curriculum in terms of mutations. So instead of putting in glutamic acid, valine is substituted instead, and then that causes the hemoglobin molecule to have a different shape, and then will form kind of a sickle shape and can't carry the oxygen. So people with sickle cell anemia have difficulty carrying oxygen to their cells, so they would be fatigued and all of the symptoms of anemia. So the process of genetic engineering is to insert, remove, or modify genes in a genome. And we can use bacteria to express genes we want translated. And the example, again, that used in biology is used again in chemistry. Insulin would be an example. So sometimes if you're using bacteria have these small circular pieces of DNA that are much smaller than their genome. They're called plasmids. You cut that with restriction enzymes, and then you cut out the human insulin gene that you want inserted, and the base pairs will match up because the restriction enzymes cut at specific spots, and then they are glued together with DNA ligase, and then you have this genetically modified bacterium. And all of them can produce the insulin protein. So they're pumping out the protein, then you ferment that, you purify it, and then you can use the insulin and distribute it to the people who need insulin to survive. So we can also genetically modify animals. So scientists originally modified the genomes of animals to study disease. So let's say modifying the genome of a mouse so that you can study cancer, make the mice more susceptible to cancers, and then you can then try to treat the mouse to see if a chemotherapy treatment works to treat the cancer, and so on. There are some other examples of genetically modified animals, and they are some pigs have had roundworm genes introduced into them to make fats that are higher in omega-3 fatty acids, which is a much healthier fat, and that makes it better for your heart health. They've also tried modifying cows to produce human milk for babies who can't breastfeed. We could also genetically modify foods. So plants can be genetically modified to make them hardier, could make them drought resistant. You could make them more nutritious. Some plants have been genetically modified so that they're resistant to a certain pesticide. So Roundup Ready crops, you can spray the pesticide on the crop and it'll destroy, or even an herbicide. So you can destroy the weeds, but then the crop stays. So there's lots of different things. Golden rice is an example that we covered in the vitamins lesson as well.